Okay, now we're going to travel from one end to the other of the North America, and we are going to hear from Guillaume Charron from Laval in Montreal.
Hello, everyone. So today we're going to talk about hybrid speciation in a unicellular microorganism. So hybridization, what is it? It is a super powerful mechanism for the generation of new biodiversity in a short amount of time. Because you don't need for two species to diverge, you just mix two already diverged species and form an entirely new one on the spot. And hybrids are really important scientifically for the study of hybridization, reproductive isolation, and speciation. Historically, because in the history of a lot of species, there are evidence for hybridization. And economically, as most of the crops we're using are hybrids between domesticated and archaic species. But when you look about hybrid species on Wikipedia, you don't find any evidence that it exists in unicellular eukaryotes. So every example you find is animal, plants. But there is no reason why they shouldn't be able to do hybrid speciation, because they have means of sexual reproduction, or at least genome recombination. And they are large populations which are not limited in their dispersal. And also there is evidence that yeast already hybridize in artificial environments, such as brewing and other kind of fermentations. And seeing the number of people at the bar every night at this meeting, I can tell that you pretty appreciate the work they do for you. So these strains particularly are involved in the fermentation of wine, lager beer, and champagne. The three main criteria for hybrid speciations are first, genomic evidence of hybridization. Two, the hybrids must be reproductively isolated from its parental lineages. And the third condition is there must be a genetic evidence that hybridization led to reproductive isolation. Our system study is the yeast Saccharomyces paradoxus. And this is the distribution of this yeast in northeastern North America. So there are two main lineages, SPC, which is found mainly in our region, so the St. Lawrence Valley in Quebec, and SPB, which is found pretty much everywhere else in North America, so from the East Coast to the West Coast. Doing population genomics analysis on those on strains we had at the lab, we found out that there was one cryptic lineage which was nested within this SPC phylogeny. And this monophyly suggests that there is already hints of reproductive isolation that could be found within this clade. This is what their genomes looks like. So this is only a SNP alignment of what is different between SPB and SPC. So in the inner circle, you find 89 SPB strains. The outer circle is 39 SPC strains. And in the middle, you find the SPC strains which are introgressed. So this is the little lineage which is found within SPC. As you can see, those strains have blocks of sequences that belongs to the B group, which indicates that there might be the product of a hybridization between SPB and SPC. There is also a private sequences which belong to only one or two strains, and those strains mostly come from the same area. So there might have been either more than one hybridization event, or there are different selection regime which could explain those patterns. And where do we find them? Well, we find them exactly where we would expect them, in the sympatry area between SPC and SPB. So, and this is like right where my university is located. This is kind of lucky, I guess. So I think we have pretty good evidence that there could have been hybridization and the genome of this Saccharomyces lineage. And next, we needed to find if the hybrid is reproductively isolated from its parent. How do we test reproductive isolation in yeast? Well, the easiest way would be to test post-zygotic reproductive isolation, which is done by mating two different yeast species, then putting the diploid into harsh conditions, so starvation conditions, which will lead to meiosis. And then we just dissect the spores which come from the diploid, put them on rich medium, then count the viable spores. So when we have high reproductive isolation, we would have low spore viability, and vice versa. Doing this, we tested a lot of crosses, around 160. On the first row, what you see is selfing, so a strain with itself. So in this case, you would expect no reproductive isolation, which is about what we see, so very high spore viability. On the second row is outcrossing, so when you cross individuals of the same lineages together. We expected to see a little bit of reproductive isolation because strains come from very different areas in North America. So what happens when you mix SPC star with its parents? Well, we have about the same level of reproductive isolation than when we mix the two parents together, B and C, which are really strongly reproductively isolated. We also found that SPC star, so the introgress lineage, share phenotypes with both its parents. So at high temperature, it grows very well like the B lineage. But when we submit it to a freeze and thaw survival assay, its survival is very low like the SPC lineage. So this could mean that it could maybe live in a different environment than both its parents. So those two things make us think that those hybrids or hybrid species are reproductively isolated because they have post-zygotic isolation and might not live in the same areas than their parents. The third thing we need to do was to find if hybridization led to reproductive isolation. This was more tricky, but we think we find at least some hints of it. Looking at SPC's genome, we found out that there was a translocation between two chromosomes, the chromosome 6 and the right arm of chromosome 13. And in this figure, what I show is the sequencing depth along the chromosomes. So you can see that we have good coverage for the region which is linking both chromosomes together within the SPC star lineage. But we don't find that a fusion event nowhere else, besides in 3 strains of the SPB lineage. So we think that those 3 strains might have come from the ancestor which maybe created this SPC lineage. And we could confirm this with the karyotypes. So with yeast, you can easily migrate the chromosomes on PFG gels, so like you would with a PCR. And we see that the chromosome 6 for all SPC star strains and SPBF strains is missing, which is explained by its fusion with chromosome 13, which is bigger. So the line that's missing, it's probably lost in the bunch of line in the middle there. And this is the same for chromosome 13, which is now smaller, so it will migrate further down the gel. And to see if this could be linked with reproductive isolation, we did a spore sequencing experiment. So we made crosses, 8 crosses between SPC and SPC star strains. We pooled the spores in different categories corresponding to the number of spores per tetrad. So if we had one spore, we would pull all the one spore tetrad together and sequence them. In the end, we had 25 pools, so 8 times N3s, and plus 1 and 4, because there were not a lot of tetrads which were fully viable in those crosses. So what we would expect if the translocation led to reproductive isolation is when meiosis happened and there is missegregation of some chromosome. Let's say that the one which is missing, the bright green part of the chromosome 13, is not viable. We would expect then to see an overrepresentation of the part of chromosome 13 which is fused with chromosome 6. And this would be translated by a bias in segregation of this part of the chromosome, and we would see it as a higher ploidy number for this part of the genome. And this is exactly what we saw. If you focus your attention on chromosome 13, which is indicated in Roman number, you can see that the coverage for the part which is fused with chromosome 6 is higher than that of the rest of the genome. And this ploidy is also correlated with the number of spore per tetrad. So the more normal number of spore per tetrad we add, so 4, the more closer we are to normal coverage. So we really think that we reunited all the conditions to identify this event as an event of speciation by hybridization. And we think also this is one of the best documented example of this mechanism. This is the biogeographic scenario that we made. So B and C probably diverged during the last glaciation when they were separated in different glacial refugium. And then were brought back together after the last glaciation, so about 10,000 years ago, which tells us that this speciation event happened really fast after the last glaciation. As genomics advances and we have more and more sequences for the genomes of fungi and other organisms, we find that there is a lot of evidence that hybridization took place in a lot of fungi lineage. And also the whole genome duplication in Saccharomyces and in yeast is thought to be linked to hybridization. So this makes us wonder whether hybridization is one of the major diversification mechanisms in fungi. I'd like to thank Jean-Baptiste Leduc and Lou Nieli-Thibault particularly, which are co-first author with me on this paper, and all the people from Graham Bell's lab and Chris Hittinger's lab which participated to this. Also, I want to thank the funding organisms and GSA for providing me with a travel award. So I'm ready to take your questions, if you have any.
We have time for a couple of questions. I have a question.
Yeah.
So you started out by talking about these yeasts and brewing.
Yeah.
So are the yeast strains that you're looking at isolated from brewing, or are they from the wild?
Yeah, I should have mentioned that. They were isolated from the bark of trees, so totally wild strains.
So totally wild
Yeah.
strains. And do you know if when you look at strains used for brewing, you said that they tend to be hybrids. So is there an idea?
Well, this is the strains which are used for brewing.
Yeah.
Yeah, there is evidence that there is a lot more hybridization in brewing strains than most of natural settings.
So there's some. So what do you think? About the why that might be?
More?
Yeah.
Well, they are all in close proximity in those kind of environments, so the chances for exchanges of material or just mating are probably higher than in nature when they probably are on 2 different spots on the tree and they don't migrate a lot.
Okay.
Thank
Is there any other questions?
Thanks, guys.
Okay, thank you.